going to discuss about cell membrane. So here we are going to detailedly discuss about cell membrane. What is cell membrane? Cell membrane is the outermost living membrane of a cell. In animal cells, that is outermost living membrane of the cell. In plant cells, that is a membrane which is living membrane which is present between the cell wall and the cytoplasm, and that separates the extracellular environment from the intracellular environment. And cell membrane is selectively permeable in nature, as it is selectively permeable in nature. It comprises of the following structure. As you can see, there is a brief look of how does a cell membrane look. After which, this cell membrane is also known as plasma membrane, plasma lemma, cell surface membrane, fluid mosaic membrane, phospholipid bilayer, lipid bilayer, phosphoglycerate bilayer, lecithin bilayer, as well as it is also known as cell surface membrane. These are all names of the same thing, which is cell membrane. And if the cell membrane is over the cell, then it is known as cell membrane. If it is on the cell organelle, it is known as unit membrane. If it is on the nucleus, then it is nuclear membrane. All of these are having same things. And what is the thickness of cell membrane? It is 7 nanometer. It is 7 nanometer, and cell membrane basically it is present in all living cells. It is present in all living cells, which involves prokaryotes as well as eukaryotic cells. And those must be living first, while these are present in all living cells. These are actually movable. These are movable, and these are foldable. They can fold themselves and they can repair themselves as well. So these are movable, foldable, repairable. After which we are having a basic composition of cell membrane. So composition of cell membrane is such that it comprises of 50% lipid, 50% of protein, whereas 5 to 10% of glycolipid or glycoprotein, glycolipid or glycoprotein. There is an MCQ point that what is the composition of RBC's plasma membrane. RBC's plasma membrane is composed of 52% protein, whereas 40% fat. RBC's plasma membrane consists of 52% protein, 40% fat. That is an MCQ point. After which, let us discuss about first the structure of plasma membrane. So, plasma membrane is basically having such a structure in which the upper layer is known as the phospholipid bilayer. The upper layer, as you can see, this structure is quite complicated and seems quite ununderstandable, but we are gonna make it quite simple. So for that, so first, I would like to show you two models of cell membrane. Two models of cell membrane which were proposed in the previous time. In the previous time, the first model of cell membrane which was proposed was sandwich model. Sandwich model or Danieli model, which was proposed in 1935, was a model of cell membrane which described that cell membrane is like a sandwich, and the upper surface layer of the cell membrane is made up of globular protein, whereas the downward surface layer of the cell membrane is also made up of globular protein. And within that globular protein membrane, there is the phospholipid bilayer membrane, and that is arranged inside those globular proteins as if it is a sandwich. So that is how this model was described, and that was also known as unit membrane model. Unit membrane model. But here, this model was not accepted, even though this model described that there is phospholipid bilayer which contains hydrophilic head and lipophilic tail, and non-polar tail and polar head, as well as the tail. They also described that it was a selectively permeable membrane, and the head was made up of phosphate, tail was made up of lipid. So even though many of the things were correct, but there were some factors due to which it was not accepted. First factor was that asymmetry. That it didn't describe the asymmetry of cell membrane. As we know that cell membranes are asymmetrical, which means if we are cutting the cell membrane in half, then both of the half will not be identical to each other. So this is known as asymmetry, and cell membranes are asymmetrical. Afterwards, it also described wrong, or we can't say it as wrong, but it described protein composition. It described protein composition as a membrane. It described the protein composition as a membrane, and thirdly, there was lack of evidence for all of this. All of this. I mean, therefore, this sandwich model, there was lack of evidence that such a globular protein membrane exists over the phospholipid bilayer and downward the phospholipid bilayer. So there was lack of evidence as well. After which, we are having what? After which, there was another model which was proposed by two scientists named and Singer, Nicholson and Singer in 1975. Now, according to this model, they said that cell membrane is basically what? Cell membrane is just like some icebergs which are floating in the sea, and they resemble the icebergs. They resemble the icebergs as mosaic. They resemble the icebergs as mosaic, and these mosaics were equal to the protein molecules. Were equal to the protein molecules, fluid, whereas the sea in which the icebergs were traveling will be resembled with the fluid which is present inside the plasma membrane, and that fluid inside the plasma membrane is considered as what? It is considered as the phospholipid bilayer. That would be considered as the phospholipid bilayer, and that phospholipid bilayer is in fluid nature. That is why we would consider it as fluid. After which, what we are left with it is that let us now discuss the structure of phospholipid. That how this structure is formed. Basically, this is the structure of cell membrane, in which there are these phospholipid bilayer. This is basically a complete phospholipid bilayer, which means it is a single layer which is made up of two layers. bilayer, bi means two, whereas layer means boundary. So here phospholipid bilayer are present in double quantity. So here the upper round part is known as the head of the phospholipid layer. Here the upper part is known as the head. This upper part is known as head. Let me change the color. So is known as this upper part is known as head. This upper part is known as the hydrophilic polar or phosphate head because it likes water, it likes polar substances, and that is have made up of the phosphate group. That is why we call it as phosphate group. That's why we call it as the phosphate head, or we call it as the hydrophilic substance. That is called as the hydrophilic head, phosphate head, or polar head. After which, on the downward side, we are having over this downward side, we are having the we are having this tail, the yellow color, light yellow color tail, and that light yellow color tail is made up of what? This light yellow color tail is made up of lipid. And what kind of lipid it is made up of? It is made of mainly made up of unsaturated fatty acids, but saturated fatty acids are also present, which cause fluidity of the cell membrane. Now let us discuss the structure of the cell membrane in detail. By the way, this lipid part of this phospholipid is lipophilic, which means it is hydrophobic in nature and non-polar in nature. It is hydrophobic, non-polar, lipophilic, as well as lipid consisting part of the phospholipid membrane, which means it is non-polar in nature, it is hydrophobic in nature, it is lipophilic in nature, as well as it is having a lipid constitution, due to which we call it as lipophilic, hydrophobic, or non-polar tail, as well as we are going to further describe that as well. First, let us discuss about the structure of phospholipid, how this structure of phospholipid is actually made. So here, first of all, as you can see over the screen, we have got here we have got a structure of phospholipid bilayer. This upper part, this upper blue color part is known as choline. This upper blue color part is known as choline. This upper blue color part is known as choline. Is known as choline. Whereas the downward yellow color part is known as phosphate group. Now, choline and phosphate group are collectively bonded together in order to form the phosphate head. Choline plus phosphate group is equal to the phosphate head, which is hydrophilic, polar, as well as lipophobic in nature. After which we are having on the downward side. On the downward side, we are having the over the downward side we are having this green color part. What is this green color part? This green color part consists of carbon hydroxyl group over this side, hydrogen group over the downward side, and another hydrogen group over the left side. So this part is basically the bond which is present between the phosphate head and the lipophilic tail. The phosphate head and lipid tail is connected by which part? This part, which is forming carbon with hydrogen atoms as well as hydroxyl group. So this part forms a bond between phosphate head and lipid tail. After which we are having what? After which we are having finally our lipid tail. And now this lipid tail is made up of what? Basically, the lipid tail is made up of two molecules, two structures, which is known as the alcohol structure, which is not clearly shown here. So here there is the alcohol structure. I guess it is present here. So it is the alcohol structure, alcohol on one hand side, and on the other hand side we are having carboxylic group. We are having alcohol structure here. This is alcohol, and the upper part is known as carboxylic group. Upper part is known as carboxylic acid. Now this carboxylic acid and alcohol group is joined together by dehydration synthesis. In dehydration synthesis, the condensation takes place, the removal of water molecule takes place to form a new compound. So here what happened? A dehydration synthesis takes place, due to which the carboxylic acid and alcohol group combine together in order to form an ester bond. And that bond was known as ester bond, and the formation of ester bond is called as esterification. Formation of ester bond is known as esterification. Formation of ester bond is known as esterification, in which dehydration synthesis of carboxylic acid and alcohol takes place. In which what happens? One hydroxylic group is taken from carboxylic acid, whereas hydrogen atom is taken from alcohol. Why? Because carboxylic acid is acid. That is why it has no work of hydroxyl group. So hydroxyl group will be taken. On the other side, alcohol is having basically basic nature. So due to this, hydrogen atom is not suitable for alcohol, due to which hydrogen atom will be lost from alcohol, and that hydrogen atom and hydroxyl group will collectively form water molecule. After which we are having what? The structure of lipid tail. This lipid tail is connected with the phosphate head with this structure of carbon, hydrogen, and hydroxyl group. And there are two lipid tails for one phosphate head. There are two lipid tails for one phosphate head. There are two lipid tails for one phosphate head. There are two lipid tails for one phosphate head. After which we have to discuss the structure now here. So here, what we are having is that we are having first of all, we are having the phosphate head, which is hydrophilic in nature, polar in nature, lipophobic in nature, which means it loves water, it hates lipid, it loves polar molecules as well, polar substances as well. On the other hand side, we are having what? On the other hand side, uh, we are having the we are having this structure which is known as the lipophilic tail, and this is made up of lipid, so it is lipophilic in nature, hydrophobic in nature, as well as this structure is lipophilic in nature, hydrophobic in nature, and non-polar in nature, which means it doesn't like non-polar substance. It doesn't like polar substance. It likes non-polar substance. It doesn't like water molecule. It likes lipid molecules. It doesn't like water molecules, but it likes lipid molecules. And here, this kind of position, this kind of structure. So suppose here, let me just write it down for your feasibility. That in this kind of structure, whenever there is a polar bond, whenever there is a polar plus non-polar bond, then this type of bond which is formed is known as between a compound known as amphipathic molecule. Whenever a polar part whenever a polar substance forms a bond with a non-polar substance, then this type of bond which is formed between the polar and non-polar substance forms a molecule known as amphipathic molecule. And the structure of this phospholipid, which I have discussed here, is known as lecithin. This structure of phospholipid is known as lecithin. So we can also say that phospholipid is having the structure of lecithin, which is having the phosphate group plus choline as the phosphate head, whereas the carboxylic group plus alcohol by the process of esterification as its tail. And in between, we are having carbon, hydrogen, and hydroxyl group as its connectivity, as its joining bond. Afterwards, we are having what? Afterwards, we are having some integral proteins inside it. Here you can see that there are some proteins inside it, which are of two types. First protein is known as integral, second protein is known as peripheral. Integral protein is also known as intrinsic protein, tunnel protein, as well as transmembrane protein, whereas extrinsic protein is also known as peripheral protein as well as surface protein. Now here, as you can see, there are two types of protein. One protein is fully embedded. Here, one type of protein is fully embedded. For instance, this protein is fully embedded within the cell membrane. It is integrated inside the cell membrane, to which it is known as integral protein, intrinsic protein, transmembrane protein, or tunnel protein. Afterwards, we are having protein which is arranged by site. That is arranged over the surface, that is not fully embedded, this is partially embedded. This is known as peripheral protein that can be easily removable. This is loosely attached, whereas integral protein is tightly attached. And these are all examples of globular proteins. These are globular proteins. So peripheral protein is also known as extrinsic protein or surface protein because it is present on the surface. And what is the function of peripheral protein? Peripheral protein is having its function to work as a receptor as well as to work as an enzyme, but it has no function in transport. As a receptor, it basically detects chemical messengers, hormones from outside the cell. Wherever there is any chemical message, whenever there's a secretion of hormones, so these hormones are detected by the peripheral proteins in order to detect what type of message do they want to bring to the cell. As well as they also form bonds with glucose in order to form glycoprotein, and then glycoprotein also acts as a receptor. Actually, they form bonds with glycoprotein in order to in order to act as a receptor, and that receptor allows to detect chemical messenger hormones from outside the cell. So these are mostly present on the outer part of the cell membrane, on the extracellular part of the cell membrane. On the other side, we are having integral protein. Integral protein is having majorly two functions. Integral protein is further divided into two types, which includes channel protein and carrier protein. That is divided on the basis of the fact that integral protein helps in transport in actual. So as it helps in transport, let us first describe what is transport in actual. How many types of transport are there? So there are two types of transport. As you can see, the first type of transport, I guess it might be visible somewhere here. Here we have two types of transport: long chain transport, short range transport. Long range transport is known as is done by xylem, phloem, as well as transport system in body. Short range transport is done by processes such as diffusion, osmosis, and active transport. Diffusion involves two types, which is simple diffusion, facilitated diffusion. Simple diffusion involves simply the movement of gas molecules, the movement of substances from higher concentration to lower concentration. As you can see in this diagram, the movement of higher substances from molecules from higher concentration to lower concentration takes place. While this is the example of simple diffusion, which doesn't require any sort of energy or doesn't require any sort of adaptability. So simply the molecules go from higher concentration to lower concentration. On the other hand side, we are having facilitated diffusion. Before discussing facilitated diffusion, I would like to discuss what is osmosis. Now, osmosis is the movement of water molecules from the region of its high water concentration to the region of its low water concentration through a partially permeable membrane. And this can also be defined as movement of water molecules from low solid concentration to high solid concentration, or from hypotonicity to hypertonicity through a partially permeable membrane. Now, if water molecules are, if a cell is placed inside, uh, for suppose if a cell is placed inside a hypotonic condition, then inside the hypotonic condition, what will happen is that cell is hypertonic, whereas the water, which is the environment in which it is present, is hypotonic. So water will move from where? Water will move from its hypotonic condition to hypertonic condition. Water will move from hypotonic condition to hypertonic condition, from outside the cell to inside the cell. So this process will be called as endoosmosis, as water moves from the extracellular environment into intracellular environment. After which we are having another condition which is known as exoosmosis. In exoosmosis, for suppose we are putting a cell inside a solution which is hypertonic, whereas a cell inside would be hypotonic. So what will happen here? So in this case, the water from the cell will go outside the cell through the cell membrane, due to which this process will be called as exoosmosis, and water will be transferred from the intracellular environment to extracellular environment. So this process is known as exoosmosis. So exoosmosis and endosmosis are these two processes, after which we are having the concept of diffusion. Here we have the concept of facilitated diffusion. Also facilitated diffusion, it is the passive movement of molecules or ions across the plasma membrane by means of a transport protein in the plasma membrane. So there are two types of transport protein: integral protein known as carrier protein and channel protein. Here there is something wrong with the definition, because facilitated diffusion is passive as well as active. It depends on the type of transport which we are having, passive and active, which means if there is the movement of molecules in particular direction, so we will be classifying it then as passive or active. So first let us discuss about what is channel protein. Here, as you can see, this protein, which is quite blurred, this green color protein is known as a channel protein. This protein is known as channel protein, and that protein is having basically a tube, a pipeline through which molecules can transfer. Why is it needed? It is needed because molecules cannot transfer directly from the cell membrane, because cell membrane is lipophilic. As it is lipophilic, many molecules such as the hydrophilic molecules, polar molecules, large molecules, as well as some ions can also transfer from the phospholipid bilayer, because it is having the lipophilic tail part, which will not allow the hydrophilic polar large molecules and ions to be transferred through the cell membrane. So for that we are having facilitations of carrier protein and channel protein. Here, channel protein is made up of eight molecules of aqua protein, known as, here it is made up of 8 molecules of aqua protein. Aqua protein is aquaporin, sorry. So here we have 8 proteins of aquaporin, and these eight molecules of aquaporin are also known as water channel proteins, which are required to form the channel protein. And that channel protein will be doing what? The channel protein will be allowing water molecules to come inside. And as I discussed, that peripheral protein acts as an enzyme. It acts as an enzyme in order to activate the channel protein as well as the carrier protein to transfer the material inside it, to utilize energy in order to transfer the material inside it. It also the peripheral protein also acts on the protein pump in order to uh, signal the protein pump to activate it in order to transfer material inside the protein pump from low concentration to high concentration using the intake of energy using the active process. So here what happens, molecules will come after the activation of these channel proteins, molecules will come and molecules will be taken inside the cell. Now here when molecules come inside the cell, there are two types of movement. There are three types of movement. There are three types of transport, which is uniport, symport and antiport. First transport is known as uniport. Uniport is a transport in which a single substance at a single time is moved across the channel protein in a single direction. Then this type of movement is known as uniport. And uniport is done through the channel protein, and as it is done through the channel protein which is facilitating the uniport is known as uniporter. So the channel protein which is facilitating is known as uniporter, whereas it is a passive process, which means no intake of energy is required. Secondly, we are having symport or co-transport. Symport or co-transport, which is present on the B part, is having two molecules. First molecule is in blue color, second molecule is in red color. Both molecules at the same time are entering across the channel protein in the same direction. So this time they are entering in the same direction. They are entering in the same direction at the same time together. So this type of movement is known as the symport or co-transport. This type of movement is known as symport or co-transport, and the channel protein which is causing or assisting the symport or co-transport is known as symporter or co-transporter. That is an active process which requires energy. On the third side, we are having this antiport. What is antiport? Antiport is the type of transport in which one of the basically this is the type of we can say this is a type of transport in which one molecule which is present in red color will be going in one direction, while other molecule which is present in the blue color will be moving in the opposite direction. So two molecules are traveling in opposite direction across the channel protein, and this type of transport in which the molecules are traveling in opposite directions is known as the antiport. Here antiport is done through active transport. It requires energy, ATP, and that actually works in active transport. It actually works in the protein pump. In protein pump, we are having this type of antiport transport, and protein pumps facilitate antiport transport, due to which protein pumps are also known as antiporter. Antiporter, I guess it's hard to write over here, so it is known as antiporter. This is a basic concept about it. And in facilitated diffusion, if it is a uniport transport, then the facilitated diffusion is a passive process. If the facilitated diffusion is having a symport transport or a co-transport, then this would be considered as an active transport or active process. So here we are having, heading back to our slide. As you can see that here, if it is uniport transport, then simply passive and passive process, no energy required. If that is symport transport, then energy is required. Both molecules will be taken inside the cell, inside the channel protein, at the same time in the same direction, which will require energy, of course. On the other side, we are having carrier protein. What is carrier protein? Carrier protein basically catches the molecule, and after catching the molecule, it rotates itself. It rotates itself on the opposite side. As it rotates itself on the opposite side, it basically throws that molecule on the opposite side of the cell, and that also requires energy. So whenever carrier protein is being utilized, carrier protein always requires energy. So Ka protein always requires energy. It is a requiring an active it is an active process. It is requiring an energy. And the movement of protein inside the cell membrane, the movement of protein inside the cell membrane is lateral movement. Movement of phospholipid bilayer inside the cell membrane is basically flip-flop movement. Flip-flop movement means if this is the phospholipid part, for suppose I guess this is a phospholipid part. If this is a phospholipid layer, this phospholipid layer will exchange its position with its opposite side. This phospholipid layer will exchange its position with its opposite side, due to which this type of movement will lead to flip-flop movement, and this flip-flop movement allows movement of molecules. On the other hand side, we are having active transport. In active transport, we are having anti-abort movement, in which one molecule goes in one direction, other goes in another direction, and that requires energy, intake of energy, ATP, and that was a good example of sodium potassium pump. Inside sodium potassium pump, what actually happens is that sodium goes outside, potassium comes inside. Utilizing one ATP, three potassium ions can be taken inside the cell, whereas two sodium ions can be taken outside the cell. So this is how sodium potassium ion works. Sodium potassium pump works. This was an MCQ point. After which we should briefly close it. And here we are having some filaments of cytoskeleton down towards the inner part of the cell membrane. We are having filaments of cytoskeleton. These filaments of cytoskeleton basically provide support to the plasma membrane. So these filaments of cytoskeleton are present on the inner side of the plasma membrane, which provide support to the these filaments of cytoplasm are present on the inner side of plasma membrane, which provide support to the plasma membrane in actual. Afterwards, we are having glycoproteins. If the carbohydrates are attached with the peripheral protein, then this is known as glycoprotein. If the carbohydrates are attached directly with the phospholipid bilayer, then this is known as the glycolipid, both having the same function to detect chemical messages to act as receptors. On the other hand side, these are examples of globular protein. And what does a fluid mosaic membrane says? This is the structure of fluid mosaic membrane, in which the protein are drifted a bit sideways, as well as one more thing which is remaining to be discussed here is that inside this structure we are having cholesterol molecules. These cholesterol molecules act as stabilizer, acts as buffer in order to stabilize. These cholesterol molecules acts as buffer in order to stabilize the fluidity of the cell membrane, as well as these prevent the entry and exit of polar molecules and ions directly into the hydrophilic, or in simple words, that prevents the entry and exit of polar molecules and ions into the phospholipid bilayer. So that is how it works. After which we got to discuss some important points. That's why then this phospholipid bilayer is basically repairable, and how it is repairable. This is because of its dual. This is because if we are eradicating one layer, then the other layer will automatically be formed, because the lipophilic tail cannot survive without its opposite layer. So that is why it is always a bilayer. Phospholipid is always a bilayer. If you are putting that in water, so the single layer in water, then other part will automatically be covered with the oppositely covered phospholipid bilayer, because the lipophilic tail would be basically not able to survive inside the water, due to which it will automatically create a phospholipid layer of its own in the opposite direction. After which there's a mechanism of fluidity, in which if they are saturated hydrocarbons, then there are more tightly packed bonds. As the saturated hydrocarbons are having very strong bond, they would be tightly packed, due to which there would be less space, due to which there would be less fluid. As there would be less fluid, there would be more solidity, due to which there can be more blockage of materials, and in several conditions that can form crystals inside the cell membrane and cause blockage of material as well. Unsaturated hydrocarbons are having weak bonds, due to which they are loosely packed and they have more space, due to which there's more fluid, due to which there's more fluidity, and as there's more fluidity, there's more flexibility, due to which mostly in cell membrane we are having most of the unsaturated fatty acids rather than saturated fatty acids. So this was a concept a bit about fluidity and cholesterol, and hopefully you enjoyed the video. By next we'll be discussing something new with a new concept. Have a good day. Thank you.